So we have different types of cells that have different shapes. Some cells are red blood cells. They are discoid, biconcave, and you have some nerve cells that are stellate. You have cuboidal and columnar cells that represent our epithelial tissue attached to the base membrane, and you have striated skeletal muscles that are fusiform. Let's begin with the cell structure. We said we divide the cell structure into three compartments. The plasma membrane, the cytoplasm, and what is the last compartment? I'm pressing it. There we go. Okay, so the cell structure is divided into three compartments. There's the plasma membrane, the cytoplasm, and the nucleus. The plasma membrane provides structural support, communication, and cell identification. And we learned about the glycocalyx, that at the surface of the plasma membrane, you have this very important layer of sugars that are attached to proteins known as glycoproteins and also glycolipids. And what is that known as? Glycoproteins and glycolipids? What do we call the glycoproteins and glycolipids? The set of glycoproteins and glycolipids on the surface of the cell membrane, the glycocalyx. Excellent. And that is important for cell identification and immunosurveillance. Your immune system is able to detect that these cells at the surface of your tissues, they have a particular set of lipoproteins and glycoproteins and glycolipids represented by the glycocalyx. And the glycocalyx is what your immune system uses to decipher self from non-self. The plasma membrane separates the inside of the cell from the extracellular space. The plasma membrane separates the fluid spaces. So there's an intracellular fluid known as the cytosol. And we said that this cytosol structurally is it a solution, suspension, or a colloid, or a emulsion? What is the cytoplasm? Is it a solution, suspension, colloid, or emulsion? Yeah, the cytoplasm, the cytosol is a colloid made up of proteins and dissolved in solution. And there's the intracellular fluid or the cytosol, and then there's the extracellular fluid. Now we're going to look at the components of the cytoplasm. It has the cytosol that we named as a colloid, that is whitish, yellowish substance. And then there are the organelles. You see the Golgi body, the endoplasmic reticulum, and the mitochondria, and you have your peroxisomes and your lysosomes, and all of these important organelles. Cytoskeleton is there for structural support, and it is the skeleton of the cell. That's why they call it the cytoskeleton. It is made up of structural proteins like microtubules and microfilaments, that enable the cells to move. So the cytosol represents the intercellular fluid and the fluid part of the cytoplasm. It is mostly water with dissolved solutes. It stores materials and is the site for some chemical reactions. Like, for example, the metabolism reactions happen in your cytoplasm, breaking down carbohydrates to get ATP that takes place in the cytoplasm. The molecular machines are in the cytosol. All these ribosomes that translate for us proteins, all the proteins that are peripheral proteins are moving within the cytosol. You have proteins like enzymes, protein enzymes that catalyze biochemical pathways that are very important for sustaining life for the cell, and they are happening at the level of the cytoplasm. So many of the organelles we've already discussed that many organelles are membrane bound. So the Golgi body has a membrane, the endoplasmic reticulum has a membrane, and that is what makes these organelles of the cell very distinct in their structure. And then that divides the inside of the cell into compartments, into compartment. That is the compartmentalization of the cell. So there is a region in the cell where the Golgi body is, and that is what helps us to identify the Golgi body. And then we can tell that this is the Golgi body, that's its membrane. It's important for being a vesicle maker. It creates vesicles, it accepts proteins and packages them. The cytoskeleton is composed of protein filaments. It's composed of protein filaments and it supports, maintains the shape, and holds the organelles in place. It moves the substances within the cell and the cell movement and the cell division. These the cytoskeletal proteins like the centromeres and the asters, they radiate from the polar sides of your cell and they attach to the chromosomes, and they are the ones who pull the chromosomes apart. So these cytoskeletons are important to help the cell move, locomotion, to help the cell crawl to sites where there's chemotaxis. In addition to, they are important in cell division. So cytoskeleton is important for providing the cell with the ability to divide, and it's important for giving the cell the ability to move. So for example, if I have two cells that want to divide, first of all, the centrioles are close to each other. Then what will happen is the centrioles are going to start polarizing to the opposite sides of the cell that is going to divide. Then your chromosomes are going to start to line up at the metaphase plate. And then the microtubules, the cytoskeleton microtubules, are going to start radiating away from the centriole to attach to the chromosomes. And so are these guys. They're going to radiate away to attach to the chromosome. After they attach, they move in the opposite direction. They move toward the centrioles, and that's what's going to pluck off all of these. That's what's going to pull apart all of the chromosomes. Going to pull apart all of the chromosomes. Okay, and they're going to get shorter and shorter. So that shows you at some point they moved away from the centromere, and at the other point they started moving back toward the centromere so that they could pluck off and remove these chromosomes. The nucleus. Most of the cells have a single spherical nucleus. They are surrounded by a nuclear envelope, and they contain the DNA, and the DNA is the template for most of the RNA. So now let's look at the structure of the plasma membrane. We already said that the structure of the plasma membrane is that it's a phospholipid bilayer. So you see that's the polar hydrophilic head, and these are the two tails. So the phosphorus head is polar and hydrophilic. It dissolves in water, while the fatty acid tails are nonpolar and they are hydrophobic. and they do not dissolve in water. What did we say was the backbone, the original structure of this phospholipid? It was glycerol with three fatty acids. So it's a triacyl glycerol. How did we convert it into a phospholipid?
Then you take O and OH and like put it together something like that.
So when we pull it together, we created a triglyceride. That's what happens in the food. So in the beginning, we used to look like that. Carbon, three carbons with OH. That is glycerol. That I asked about that on the lab exam. What is glycerol? And people thought it was sugar because it began with like glycoprotein and glycolipid. No, that is a lipid. That's fat. And then you have your free fatty acids. Okay, we remove that. Okay. Then they connect like this, but that's still not a phospholipid by the way. That's a tri, that's a triacyl glycerol. What happens is we remove, that's a triglyceride or triacyl glycerol. So what happens now is we remove one of the tails and we replace it with a minus phosphate and a choline. That's the head, and these are the two tails. That is what I was asking about. These are the, you see, these are the two tails. These are the two yellow tails, and this is the blue head. The blue head is hydrophilic, dissolves in water, and the two yellow tails, these are hydrophobic, and they do not dissolve in water. There we go. So these are peripheral proteins. Peripheral proteins stay on the outer leaflet or the inner leaflet, and the transmembrane proteins, or also known as integral proteins, they span both leaflets of the bilayer. On the top, you have sugars attached to proteins known as glycoproteins and sugars attached to the lipids, and these are known as glycolipids. And both of them, glycoproteins and glycolipids, are known as the glycocalyx. The fluid mosaic model is showing you that this membrane is so liquidy and fluid. What makes the membrane so rigid? Is these orange guys? What are they? Here. Here. What is that? Cholesterol. So the more cholesterol you add, what happens to the plasma membrane? It gets more rigid. So proteins, lipids, and carbohydrates are scattered in the membrane. Phospholipids and components move in the membrane. So these phospholipids, they are not held in place. They shuffle. They switch from left to right, from right to left. Some of them even flip upside down. So this phospholipid falls down and this one gets up on its place. So this phospholipid bilayer is not held in place like a fence. It is a fence, but it's a moving fence. That's why they call it phospholipid bilayer fluid mosaic model. Now let's talk about membrane proteins. Membrane proteins are either integral or peripheral. So membrane proteins are integral proteins. Integral proteins span the entire width of the membrane. You see, these are integral proteins. So you should be able to tell on the exam if I point to These structures, what are those? You can tell these are integral proteins or transmembrane proteins. While peripheral proteins, they are only on one side. They are only on one side of the membrane. What are the functions of the membrane proteins? They transport membrane proteins. They transport, like for example, bringing in glucose. These transmembrane proteins, they literally grab and bring in glucose, called facilitated diffusion. They can also be receptors. We talked about that with epinephrine. Epinephrine bind on the outside, it cannot get in, but once it bind on the outside, what happens to the transmembrane structure? Start shaking and moving, and that causes the internal leaflet to go and the internal peripheral membrane to go and propagate a signal transduction cascade like a domino effect, one after the other, that would culminate in the end in a second messenger, and the second messenger will mediate the target effect. So I cannot do the target effect on my own. Yes.
So the transmembrane protein is not the same as the integral protein?
Yes. It has two names. And peripheral proteins are on only one side, either the outside or the inside. Functions of the membrane proteins. There are also enzymes like the sodium potassium ATPase. That's an enzyme. It grabs three sodiums, throw them up against concentration gradient, or grabs two potassium and sends them inward down concentration gradient, against concentration gradient. So it's always working against concentration gradient. Structural support and linking the cells. So there are proteins like collagen, proteins like keratin. They are important for holding the cells, and some proteins are important for linking the cells together, like the desmosomes, like the tight junction, like the connexins. These, you see the cells, let's say all of you guys are cells, but you are separated. But the cells is like they are in an auditorium. They are so tight, they were in the subway, sitting in that subway seat together. In fact, not only the cells in your body are touching, there are proteins that are holding them, like stapling them together. These proteins are actually stapling them together so tightly that even water cannot seep in between. That's how tight these proteins hold and link the cells together. So, for example, you have hydrochloric acid in your stomach. If that leaks out into your intestine or into the core of your body, it's going to destroy your body. But the cells of your intestine are so linked tightly together with CAMs, C-A-M, cell adhesion molecules, or CAPs, C-A-P, cell adhesion proteins. These guys, they are linkers, and desmosomes and tight junctions. These are all examples of proteins. You see, that's the role of the plasma membrane in transport. So you have channels that allow the ions to cross the membrane. You see, this is a protein channel. A protein channel that allows the passage of two different molecules. That's not a leak channel. Remember, I said a leak channel can only allow the passage of one, either from inside to the outside or from outside to the inside. But this one is most likely gated. So it allows the exit and the entry, or it couples the passage of one molecule down concentration gradient to fuel the passage of another molecule outside the concentration gradient. So the channels allow ions to cross, channels allow the ions to cross the membrane. And the carriers bind and directly transport the molecules across the membrane. You see, this is facilitated diffusion. They bind, so they close from the bottom, open from the top, bind glucose, then close from the top, open from the bottom, and let it come right out. So transporting substances, some membrane proteins act as cell channels or carriers to transport the substances into or out of the cell. The carriers bind and directly transport the molecules across the membrane. The carriers bind and directly transport the molecules outside the membrane. The receptors bind the chemical messengers called the ligand. So you see, these are ligand gated. We talked about those. Ligand gated bind the outside, and then upon what's the name of the molecule that is ligand and it binds on the outside, leading the channel to become open and to allow for the influx. Jessica, acetylcholine. Excellent, 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 excellent. Okay, I'll give you 1 point extra on the exam. Excellent, excellent, very excellent, excellent. Okay. I said this many times, but you have to always, you're gonna flip through your page, but it's not an open book. So you gotta flip through your mind. So you gotta go home and reread so stuff can go from the book inside the mind. It's not gonna flow with diffusion from the book into your mind. You have to read it, read it, read it. So epinephrine would bind a receptor, and the receptor would mediate an intercellular signaling. But that's not what we're talking about. We're talking about a ligand that binds a channel, and once it binds it, the channel would open and there will be influx. You see, once the orange ligand acetylcholine binded here, it opened and all of the substance flew inside. Okay, so acting as a so some membrane proteins act as receptors binding to a ligand to trigger a change in the membrane protein or the cell. So the ligand binding produces changes within the cell. Okay, enzymes. Enzymes speed up the chemical reaction. So many are associated with the membranes. So acting as enzymes, some membrane proteins act as enzymes, catalyzing chemical reactions. Who's an example of that? You give this one, I give you another point. Jessica. What's an example of that? She's the only one waking up. So now she was sleeping too. So somebody else now gotta help me. What is the name of give me an example of this. It just said enzyme. Give me an example of this. An enzyme that is membrane bound that will take a substrate and convert it to a product. And please don't tell me sodium potassium ATPase, because that's the type of an enzyme that influx, efflux. It will bring back influx the potassium and throw out the sodium, efflux the sodium out. But this is an enzyme on the cell membrane that causes a product to be generated from a substrate. An example of that is what? Yeah. Okay. Jamie, what is this enzyme? I mentioned it already. Okay, so now it's gonna be half a point. I'm gonna help you. I meant this is part of the epinephrine. When epinephrine in the cascade, when epinephrine bind outside, and then it causes a peripheral protein to cause the transmembrane protein to dimerize and change, then an intercellular protein, G protein is going to move to this guy, to this enzyme, and this enzyme is going to make for us the second messenger. Catecholamine. Yeah, the catecholamine is the epinephrine because he cannot get inside. I hope I still have it up there. No, I erased it. No, I erased it. Yeah, I erased it. No, I erased it. Okay, so remember, I showed you in the cascade. What happened was, in the cascade, what happened in the cascade for epinephrine, we have our bilayer leaflet. I am tired. You have a bilayer leaflet, and you have the peripheral receptor, and then you have transmembrane receptor. Then epinephrine will bind here. Then after epinephrine bind will cause this guy to dimerize. Then it will send the peripheral protein, G protein. The G protein is gonna go to an enzyme here who will convert ATP into cyclic AMP. That is my second messenger. It's called adenyl cyclase. adenylyl cyclase is gonna cyclicize ATP into cyclic AMP. Then cyclic AMP is gonna go and activate a kinase, phenylalanine kinase. It adds a plus PI. It adds a phosphate group, and the phosphate group is going to cause the enzyme to become from off. It will become on, and then that enzyme that's on is gonna give me my target protein. Gonna give me my target protein. So that's adenylyl cyclase. That's the second messenger. That's this one. It will take, who's the substrate? ATP. Who's the product? Cyclic AMP. That's my second messenger. So that's an example of a transmembrane protein who is an enzyme. What's another type of transmembrane protein who is an enzyme? That is not enzyme to give you a product, but an enzyme to do a job. What's the name of another enzyme on the plasma membrane that is not this type of, not all the enzymes give you products. Some enzymes mediate something else, like sodium potassium ATPase. It takes the sodium from inside to the outside and potassium from outside to the inside. So sodium potassium ATPase is an enzyme, but adenylyl cyclase is another type of enzyme that gives you products. Okay. Transmembrane proteins in the cell membrane are important for structural support. They may bind cytoskeleton proteins in the extracellular fluid and the cytosol. They provide the cell shape and maintain the structure. So you see, providing structural support, and some membrane proteins provide structural support by binding to other proteins in the extracellular fluid. You see, they bind to other proteins in the extracellular fluid. Linking the cells. So these, you see, this is what I was talking about, linker cells. These are known as what? Desmosomes. That's one kind of them. And so the desmosome is the kind. Desmosomes, the tight junctions, the connexins, the gap junctions. But what did I call them? Three-letter words. What did I say? Three letters. Yeah, very good. CAMs, these are the cell adhesion molecule. What's the other one? CAPS, yes, cell adhesion protein. You see, they have cell adhesion they hold the cell together so tightly, even water cannot seep through. It's crazy how tight they hold the cells together. These guys. They also allow communication, but these are not the linker. These are the gap junctions. You see, they link the adjacent cells. Some membrane proteins link the adjacent cells together. Other membrane components like cholesterol, glycolipid, that's the glycocalyx. So you see other membrane components are like cholesterol that stabilizes the membrane, and there are glycolipids and glycoproteins outside the membrane. They function in cell recognition. And we said that these glycolipids and glycoproteins, the glycocalyx, they are important for immunosurveillance, and they give the cell its identity. Transport across the plasma membrane. So the transport across the plasma membrane, there's passive transport, does not require energy because it flows down concentration gradient. Example of that is the leak channel. So the leak channel, things are leaking down concentration gradient. Okay, the water leak from the roof because there's a lot of water in the roof, less water on the first floor. That's why it leaked down. It doesn't leak up. The water's not going to leak through the air. It's going to go from area of high to low. What about ligand gated? Is it not, again, a down concentration gradient? It is. It just was closed, the channel, but once it's open, everything is going to leak down concentration gradient. It is only the sodium potassium ATPase that is working against concentration gradient because it's throwing sodium outside the cell, and it's throwing potassium inside the cell. The active transport requires the cell to expend energy, ATP. Passive transport is diffusion and osmosis. requires a concentration gradient. So the movement of molecules from area, you see that's a higher concentration, that's a lower concentration, so they're gonna start moving out. Is driven by kinetic energy of solute molecules. The diffusion is the movement, the movement continues until it reaches equilibrium. And equilibrium is when concentration is uniform throughout the container. What are the types of diffusion? We talked about simple diffusion and facilitated diffusion. Simple diffusion is when you just go through the cell like a leak channel. Facilitated diffusion, it means you do have a concentration gradient. There is a lot more outside than inside, but you're just so big you cannot fit in. So you need a facilitator, a mediator to facilitate your passage down concentration gradient. What's an example of a facilitated diffusion? Who do we need to transport through facilitated diffusion? I'm sorry? Don't be afraid. Say it. It may be the truth. You don't even know it. Protein. We're moving proteins down concentration gradient through facilitated diffusion. We're moving proteins. Proteins do not move with a channel. Proteins enter the cell or exit through exocytosis or endocytosis. We're talking here about diffusion, things flowing down concentration. I'm sorry? Ions don't need a facilitator. They just flow through the leak channel. Remember, the leak channel open or the gated channel open, they flow through it. So now for facilitated diffusion, what is the problem with facilitated diffusion? You're so big. The issue that there is more on the outside, that's not a problem. It's already given. There is a lot of calcium outside, it's going to flow inside through the leak channel or the gated channel. More sodium outside is going to flow through the leak channel or the gated channel. Give me an example of something that is a lot on the outside, but it cannot go through the leak channel. It's so big. We need a facilitator that facilitates its passage. What is that? I said it. I said it, literally grab it from outside, bring it inside.
That,
Hmm?
the word, I can't pronounce it.
C? Cholesterol?
No.
Cholesterol, we don't need it inside. We need it in the membrane to hold the integrity, give the membrane is not so catecholamine. Catecholamines do not enter the cell. Catecholamines, you defeated the whole purpose. Catecholamines never get brought in. They bind ligand. Acetylcholine is on the outside. You guys don't understand the question. I'm asking you that if you have a membrane, and the membrane, you have leak channels to allow sodium to come in, or leak channel to allow calcium to come in. There is something else that I need to bring in, but I cannot fit the channel. I need a facilitator to grab it, bring it in. The ligand binds on the outside, guys. So acetylcholine binds on the outside, that dimerizes the receptor protein. The receptor protein sends a peripheral protein, and the peripheral protein goes into the adenyl cyclase through a second messenger. That's why it needs a second messenger. The epinephrine and the ligand, please listen. The epinephrine and the ligand, if it can get in, why does it need a second messenger? Why is the reason it needs a second? Because it cannot get in. So it stays on the outside because it's polar. It is something that on its behalf, the second messenger goes and does the job. So give me something that needs to get in from outside to the inside. I'm sorry? What happens with insulin? Insulin. What happens with insulin? Why your body secrete insulin? So that means there's a lot of glucose where? In the blood. There is a lot of glucose in the blood. Now we need to bring in glucose where?
We need it to, has to be absorbed by the cells.
Yeah, by the cells. By the cells. So you have a lot of glucose on the outside that must be brought in. So insulin, what insulin would do, it will make mRNA that when translated would give you GLUT. GLUT are the glucose transporters. So you'd have GLUT, and many these GLUTs, they are gonna grab glucose down from the blood where there's a lot of glucose. So facilitated diffusion is for glucose because he's so big and big. These sodiums are so tiny, the calcium so tiny, go through a leak channel. But there is no leak channel for glucose, it's huge. That's why we need GLUTs, these facilitated diffusion channels. I said it is so big, it literally needs a facilitator. So look, simple diffusion involves nonpolar solutes passing directly. You see, these guys are nonpolar. They diffuse so simply. You see in both directions, there is a lot of orange outside, diffuse where there's a little bit of orange. Or there's a lot of purple inside, diffuse where there's a little bit of purple. It involves charged or polar solutes crossing the phospholipid bilayer using a that's facilitated. So for facilitated, that could be for some charged molecule that cannot go through the lipid bilayer, and also with some sort of ionic compound like glucose. You know, glucose is polar. Remember when we drew the structure with a glycosidic bond, and glucose has oxygen and has all of that. Remember? Yeah. So it needs a facilitator right here. Osmosis is the diffusion of water from an area of low solute to an area of high solute. You see, if you have this barrier here and you got glucose, there is a lot of glucose on side A, a little bit of glucose on side B, but this membrane dialysis, the pores are so small. Remember, glucose is huge. So the pores are so small, water is going to start moving from an area of low concentration to an area of low concentration of glucose to an area of high concentration of glucose to dilute it, and that is known as one of the properties of water. Water has high specific heat capacity. Water is a versatile solvent. It dissolves most of the substances. So water moves from solution of lower solute concentration to one with higher solute concentration. See, you see what happened with osmosis? All of the water flew from one area to the other to dilute glucose. It is driven by osmotic pressure or the pulling force that solutes exert on the water. So when the concentration of solute reaches equilibrium, water molecules move at random, but there is no net movement. There is no net movement. Yes.
Okay, that's about osmotic pressure, so I think I misunderstand what it's there for. So is it there to stop diffusion?
So osmotic pressure is describing the driving force, the tendency. So for example, if there is 500 millimolars of glucose on one side and 100 on the other side, there is a very big pulling force, osmotic pressure. If I double it from 500 to 1000, now the osmotic pressure doubled. So the pressure is the driving force, the tendency. So that is pulling the water to move and stay at the site where, and block the water from returning because there's a lot of solute. Okay? Yeah, so driven by osmotic pressure or the pulling force that solutes exert on the water molecules. So when the concentration of solutes reach the equilibrium, the water molecules move at random, but there is no net movement. The volume changes in osmosis, and the fluid changes on each side of the container. Okay, so the volume changes in osmosis, and this is, we talked about that. That's in the tonicity. That is in the so we said if you put, so this is the membrane, the plasma membrane of a red blood cell. The tonicity compares the osmotic pressure between two solutes. If the extracellular fluid is normally isotonic to the solute, there is an equal rate of movement of water from an area of a higher constant, and it enters and leaves the cell at the same rate. So the solute concentration is equal inside and outside the cell. No net movement of water. The volume changes. So now let's talk about the volume changes in osmosis. There's a hypertonic or extracellular solution. So the hypertonic solution has more solute on the outside than inside. So think of it this way. There was an eraser somewhere. Okay, thanks. I am tired. I went to sleep at 2 a.m. because there was another class. I gave them 100 question exam, and some of them they did well actually. They got in the 90s, but a lot they did bad, and they blame me. You don't explain, you don't tutor us. You don't tutor us. I said, okay, I'm sorry. I'm gonna tutor you now. So I made like a 4 hour tutoring video, send it to them, and then yeah, it's hard. It's hard being a professor. You work hard and you get blamed.
You're teaching a hard subject.
Yeah, that's biochemistry, yeah. But you have to do your best. So when the book is closed, you have done your best. Okay. And then and students have to go home read. Some students have the attitude that their relationship with the course is the 3 hours with me now. Then they leave, they have nothing to do until they meet next week. That is what frustrates me because people gotta go home and read and study. Not every student is like that. Many students do well. But if I give you a study guide that is really close to the exam questions and we explain it on the whiteboard and we talk about it here and we record the lecture and nobody's willing to put in the work, then that is just very aggravating. So if we have 3 beakers, 1, 2, 3, and we put 3 red blood cells. On the first beaker, the osmolarity is 700 milliosmoles outside, and inside is 100 milliosmoles. So the outside is more hyperosmolar or hypertonic. Here, 100 milliosmol inside, 100 milliosmoles outside. And here it's 100 milliosmoles inside, and outside is like 10 milliosmoles. So here they are equal. That's isotonic, like isosceles triangle, isotonic. And here the outside is less. So that's hypotonic. And here it's hypertonic. So you must be able to tell which is which and the fate. If it's hyperosmolar, water goes outside. And here there's a steady state, steady state. Okay, here there is less on the outside. That's hypotonic. So what happens is here the water is going to move from outside. Water here is going to move from outside to the inside because there is more solute concentration in the inside. So this is going to cause it to swell. Here the red blood vessel will swell. Here it will stay the same, and here it will shrink. Shrink. Any questions? Okay. That's hypotonic. And okay, active transport is where we use ATP. We use ATP in order to move solutes. So proteins pump a solute against its concentration gradient using the energy from ATP. There are 3 types: the uniport, a symport, and an antiport. So the uniport pump, it drives a single substance into or out of the cell. That's a uniport. The same port drives two or more substances in the same direction. And they're also called co-transport. So they drive both in the same direction. So they drive two, that's known as co-transport. It drives two or more substances in opposite direction, and that is known as counter-transport. Active transport is either primary or secondary. It's primary because the sodium-potassium pump, also known as the sodium-potassium ATPase, the major primary active transport pump. You see what it does is the sodium in the extracellular fluid is 10 times greater than the cytosol, and potassium inside is 10 times more than outside. So the sodium-potassium ATPase maintains a steep concentration gradient by ensuring that the too much sodium 10 times outside does not equilibrate with the one inside. And the 10 times potassium that's inside does not leak out and balance with the outside. So it ensures that it is a disequilibrium. So when the action potential comes through, there is actually a gradient for the movement of these electrolytes down their concentration gradient, and that is what helps exciting the tissue and relaying the stimulus and information. Sodium potassium pump is a pump that binds three sodiums from the cytosol. An ATP is used, is hydrolyzed, and the pump releases three sodium into the extracellular fluid and binds two potassium. The pump releases two potassiums into the cytosol. You see, the sodium potassium ATPase binds three sodiums. The pump binds three sodium from the cytosol, and then ATP is hydrolyzed. The phosphate binds the pump, and the pump changes the shape. Then the pump releases the three sodiums into the extracellular space. Then the phosphate detaches, and the pump changes back into its original state. And then the pump releases the two potassium channels to the inside. In secondary active transport, here we're coupling. We're indirectly coupling. It uses ATP indirectly coupled with active transport of another substance. So an example of secondary active transport is the transport of glucose coupled with movement of sodium. So sodium moves down concentration gradient. We said that sodium, there's 10 times more sodium outside than inside. So sodium can easily flow in. So we allow sodium to flow in, and with it, while it's moving down, we couple the movement of glucose down concentration gradient with it. Glucose transport, both sodium and glucose bind a symporter, and the symporter transports sodium and glucose into the cell. A carrier protein uses the potential energy of the sodium ion gradient to power the transport of glucose. And you see, sodium is in purple, glucose is in green. So we use the movement of sodium down concentration gradient to allow for the movement of glucose. So do you see? We use the movement like you have a car, you try to go to college, somebody takes a ride with you. That's an example of what happens here. Sodium is moving down because so much sodium outside, 10 times than inside. So it flows like a waterfall. So when it's moving down, then calcium, then glucose takes a ride and flows in with it. So the glucose transport, the sodium binds to the sodium potassium ATPase primary active transport pump, and ATP is hydrolyzed. The pump creates sodium gradient to drive the glucose against its concentration gradient. You see, this is the sodium potassium ATPase, ensuring that there is a lot of sodium outside so that sodium can move down concentration gradient and carry with it glucose. So the resting membrane potential is a consequence of the ion transport across the plasma membrane. So the movement of ion produces unequal concentration between the cytosol and the extracellular fluid. The charges separate across the plasma membrane, and it is positive outside with negative inside. Active transport via the vesicles. There are larger particles transported in small sacs of the membranes. Okay, active transport via the vesicles. form by pinching off the cell membrane. That's the endocytosis we talked about. And they fuse with plasma membrane and other membrane. There are two types of vesicular transport, endocytosis and exocytosis. Endocytosis are movement of substances into the cell. Examples of endocytosis, they move extracellular fluid, moves the extracellular fluid and large molecules and the whole cells into the cell. There are two types. There are two basic types, the phagocytosis for particles and the pinocytosis for fluids. You see, now we have taken in, that's phagocytosis, we have taken in our dangerous green bacteria, and that's known as phagosome. The next step, a lysosome will come and will swallow this phagosome. And this is pinocytosis when we bring in cellular material known as um, pinocytosis. There's a receptor-mediated endocytosis, and that is a clathrin-coated pit. So you see, you can bind into a ligand. You see these receptors, you bind to them, then they will internalize in until they make a coated vesicle. In exocytosis, the opposite. We make a vesicle that fuse now with the membrane, and then they exocytose. Exo is exit to the outside of the cell. Is the reverse of endocytosis, and particles are released in exocytotic vesicles. So that's simple diffusion. When you move down concentration gradient, you don't need any help. Facilitated diffusion, you're still moving down concentration gradient, but not directly. You need a mediator. And osmosis, it is just the movement of water from an area of a low solute. You see there is a little bit of green to an area of high solute, a lot of green. And primary active transport is when the sodium potassium ATPase uses ATP in order to throw three sodiums against their concentration gradient to the outside, and they throw two potassium against concentration gradient to the inside. Secondary active transport is when you couple the movement of sodium down concentration gradient with the movement of glucose down concentration gradient. You see, the two arrows are pointing to the inside, So the particles, this is the particle, this is the receptor, and this is the pseudopodium. This is pinocytosis, and this is a protein-coated pit. And you see now, pinocytosis, we internalize, we bring in the substance, while in the receptor-mediated endocytosis, we bind to a receptor that mediates the endocytosis. While exocytosis is the reverse, where we fuse with the membrane and we throw the content out.